So for test crosses, these are just used to determine what the second allele is in an organism. And this happens when you have an organism that has the dominant trait. So when you have an organism that has a dominant trait, there's two possible genotypes. So they could be big P, as you see in the first example Punnett square on the left, or they could be big P, little p. So to figure out which type of genotype your dominant organism has, you just need to do a cross with a homozygous recessive organism. And that's what's being done in this example here. So we have big P being crossed with little p. And we have big P being crossed with little p. And so when you analyze the results of the offspring, if they're all dominant, there's a pretty good chance that your offspring is homozygous dominant. But if you have even just one offspring that's homozygous recessive, that means that your organism has to be a carrier, or it has to be heterozygous. Okay, linked genes, this is just saying that these genes are really close together on a chromosome, and when that happens, they're more likely to be inherited together because they're not going to be crossed over during prophase 1 of meiosis. So if they're close together, they will probably stay together. Multiple alleles, this is when 3 or more alleles are coding for the same trait. So the example you should know of this is blood type. So for blood type, we have, you can be either type A blood, type B blood, type AB, or type O. And there's a bunch of different genotypes for this. So remember that there is the big I, big A allele, there's the big I, big B allele, and then there's the little I allele, which is the recessive allele. And when you have two recessive alleles, little I, you'll be type O blood. If you are big I, big A, big I, big B, then you're AB blood. For just type A blood and type B blood, remember that there's two different genotypes for both of these blood types. So you can either be homozygous for type A blood or heterozygous. If you're heterozygous for type A blood, it means you're big I, big A, little I. And similar situation with B blood. If you are heterozygous for B blood, you're big I, big B with that little I allele. Okay, so this is multiple alleles. Last thing we talked about for genetics was pedigrees. So these are just used to show inheritance of some sort of trait or disease through a family. The circles are females and the squares are the males. And so you need to make sure you are good with looking at a blank pedigree and just being able to figure out which genotypes each of these individuals are. So maybe pause the video now and then make sure you can do that and make sure all of that makes sense to you. Okay, last thing we have today is biotechnology, and this is just using science and technology mostly involved with DNA and manipulating DNA to really improve life. So first we need to make sure we know that stem cells are cells that can transform themselves into any cell that our body needs. Okay, so that could be skin cell, brain cell, blood cells, fat cells, skin cells, any cells. And the other definition on here is differentiated cells. This is just saying that you can take that skin cell and differentiate it into any of those types of cells. And this is done by gene expression. So remember that every single cell in your body has the same DNA in it. You don't have random bits of DNA in your liver cells and your skin cells. It's all the same because DNA makes you you, so every single one of your cells has the same DNA in it. Makes sense. So the reason why these cells can look different is because your cells are capable of turning on and off the certain genes that they need to function. And that causes them to look a little different and allows them to be able to do different jobs. And so that's gene expression. Okay, the next thing we talked about was DNA fingerprinting. So remember we did that really cool lab where we did that gel electrophoresis, and that just separates the DNA based on size. So remember that the really small pieces of DNA can go really, really far through the gel. They're able to wiggle through there and go far. But the big, big, big pieces of DNA, they stay towards the top. And so this technique is used to solve murder crimes. You can figure out who the parents are of offspring. This example here on the slide is looking at the DNA that was found at the crime scene, and then there's two different suspects on here. So when you look at suspect 2, you can see that that matches up with the DNA that was found at crime scene 1. And so we would say, using this DNA fingerprinting technique, that suspect 2 is probably the guilty suspect here. All right, recombinant DNA and restriction enzymes. So restriction enzymes, it says here, cut DNA at certain sites. So these were the DNA scissors that cut DNA in certain places. And this is important because if we cut a source of DNA at a certain place with a certain restriction enzyme, we can use that same restriction enzyme to cut out a gene of interest, and we can then stick it into that DNA, and now we can make something like that glowfish, for example. So now that glowfish has recombinant DNA. It has DNA from a different organism. All right, this is the very last thing here. So just genetically modified organisms that we also call GMOs. So these are just organisms with modified genes. You can take a gene from somewhere else, stick it into another organism just to give it the desired trait. Or like in this corn plant here, we can make it resistant to this, what does it say? Crop is infected by European corn borer. Pest dies when feeding on the plant. So we just give it this certain gene that allows it to produce a protein that kills off this pest that otherwise would be killing the corn. All right, this concludes your DNA, your genetics, and your biotechnology review.